integrating multiscale modeling and machine learning in computational medicine. But let me mention some housekeeping issues. We continue to provide CME credits for our tech talks and our ID series presentations. We will distribute the information via email to those who registered today. Sorry. If you did not register prior to today's talk, you can still obtain CME code information from Deborah Rees, and we'll put her email address in the chat box. So some upcoming events. On April 3rd, we'll have another tech talk from Dr. Dan Harris from University of Delaware also, and we'll talk about data linkages for pharmacoepidemiology, case study on effects of shingle vaccination. So this is April 3rd, that's a Thursday, and that's at noon. And on April 22nd, we'll have an ID series, that's a Tuesday. So the tech talks are on Thursdays, the ID series are on Tuesday. And that will be from Julie McCullough, who is at Computational, as well as University of Delaware, who will talk about opportunities to advance rural healthcare using a Scottish model. So these two next presentations are really exciting, so please do not miss them. So again, I'm really delighted to introduce Dr. Deng. Dr. Deng is assistant professor in the Department of Computer and Information Sciences at University of Delaware. She had obtained a Ph.D. at Brown University School of Engineering and a Master of Science in Applied Mathematics also from Brown. Prior to coming to University of Delaware in August 2024, she was a postdoc fellow at the Ragun Institute at MIT and Harvard. She has already a very large number of publications and multiple awards. She is a popular speaker, has been invited to give talks and lectures all over the country, and notably, she was one of the panel speakers at the last 2024 Innovation Summit here at Computational, the future of healthcare. So we are really honored to have her today giving us a talk on her research. Thank you so much, Dr. Deng. Go ahead.
Thank you. Thank you, Caroline, for the nice introduction. Yeah, thank you very, very much, everyone. So I'm really happy to introduce our work on integrating machine learning and multi-scale modeling in computational medicine. Hope you can see the screen really well. So here is a very interesting sketch denoting the machine learning and also the multi-scale modeling. So maybe most of you are familiar with machine learning since now we are in an AI intense data generation like era. So machine learning has been very useful to solve a lot of data-driven problems. And well, if you go into some theory, maybe some machine learning now going to some interpretable machine learning, they will be able to help us understand the theory while it's remaining an intense area. And then also, on the other hand, multi-scale modeling has been built on prior knowledges. Here, my PhD group was focused on like physics-informed neural networks. Is also physics-informed models. For example, like molecular dynamics, anticipated particle dynamics, and then also finite element method that we developed to solve multi-scale modeling in different time and space scales. And it's built on top of our prior knowledges, so it's actually very theoretically plausible. And the reason of integrating both is that we want to develop those models, so-called physics-informed machine learning models, which can help exploiting the underlying physics while also make use of the big data that we generated by different fields. So I want to have the following disclosure and acknowledgement: no financial or other conflicting interest related to today's presentation. My work presented today has been generously funded by NIH and NSF. So the motivation during my PhD and postdoc, and also a large focus of my current lab on computational intelligence for dynamic system, is that we are interested in designing digital twins through multiple different methods. For example, the multi-scale modeling that I mentioned, which are driven by physics or chemistry. And then also there are machine learning models, as I mentioned, the data-driven models where we had the less prior knowledges, but the big data will help us reveal the patterns underlying. And then there are also other fields which is intersection of both. We are interested in developing multi-scale modeling as well as machine learning to help interrogate the system and build digital twin of either patients or other animals to help us understand mechanism, and then we can provide mechanistic insight to cure our patients. So I want to start with a brief overview of my first author work. In the multi-scale modeling regime, I have worked in developing coarse-grained molecular dynamics models for this kind of fiber assembly and testing those mechanical properties and matching them with the observation from a clinical or in vitro or in vivo experiments. On the other side, majorly of my work has been focused on dissipative particle dynamics, which is a method has been very useful in simulating a mesoscale, which is intermediate between micro scale and the macro scales. And our model developed has been successfully applied for simulating microvascular networks, for example, like the microfluidic device we developed by our collaborator. And then also our model has been employed to simulate like microaneurysms in diabetic patients that we acquire from our collaborators, these very specialized geometries. And what's more challenging in terms of computation intense is this whole blood simulation, including not only red blood cells, platelets, but as well as white blood cells. So also in the machine learning part, I have worked on developing deep learning models to help glucose predictions and also Gaussian process regressions to understanding time series data. Further down the string, I also worked on developing systems biology informed neural network, which is a particular type of physics informed neural network, combining with control algorithm represented by reinforcement learning to help patients with diabetes achieving a better glucose stability. And then during my postdoc, I've been working on developing interpretable linear models to understand high throughput antibody profile data generated by one of my PhD advisor lab, which characterize a lot of immune responses in terms of different infectious diseases. So let me talk about the multiscale modeling part, and specifically with a broader application of thrombus formation, and that's really very critical. So as you may know, thrombus formation is a very dangerous and complicated process. When it's happening in the microvascular, it will affect clog the vascular networks and even resulted in 90% of deaths in COVID-19 patients. And it can also happen in veins. And when it happens in veins, it causes a disease called deep vein thrombus, and it affects a lot of our vascular system, leading to drastic disability or death, according to the National Alliance statistics. And if we zoom in, we can find that thrombosis are actually blood clots, meaning that it consists of platelets, red blood cells, and protein. And if we further expand the thrombus formation process, we can find that it starts with aggregation of these proteins like fibrinogen or VWF, etc., on the substrate representing the vascular surface, or maybe a wounded surface, actually. And then the free-flowing platelets will then be recruited by these activated proteins. And then further down the time, you can find more red blood cells will further being recruited and adhere to this network. And then the fibrin is a downstream product of fibrinogen during this whole processes will enhance the blood clot. So you may find out there are two important factors addressing this significant process. Is that the physical, or you can say the mechanical property, because the cell motions are driven by the blood flow that's happening all the time, and also it involves with adhesion between molecules like protein with cells and cells among cells. Another important factor is the chemical factor, which are responsible for the transport of a chemical species that allowing the aggregation of these proteins and cells. However, it remains unclear how this factor holistically contribute to the thrombus formation. And that's what my motivation of designing multiscale modeling for this. So why am I interested in developing in silico models? Is that one, it can directly analyze both in vivo data and in vitro data. And also second is that when I perturbed in silico model systems by changing the parameters, I can use this well-constructed model to infer the behavior of these systems in terms of in vivo or in vitro scenarios. And third is that it can provide this in silico model by different perturbations can help us provide a causality of the system that we're interested in. So specifically, the dissipative particle dynamics models we employed in the thrombus formation are Lagrangian point view, which is mesh-free, and particle-based modeling. And then all these chemical and physical constraints, we can impose them using the chemical potentials. And specifically within this framework, every component of the system, whether the cells or proteins, are represented by particles. And these particles are subject to the three different kinds of force. One is a conservative force, second is a dissipative force, and then third is a random force to keep the whole system physically consistent. And specifically, another advantage of our simulation approach is that it's usually 3D, meaning that every particles have a 3D coordinates that we're interested in looking at. For example, I'm showing you a very interesting example of in silico red blood cells that we can represent by concave shape of red blood cells using this triangulated mesh. So every point will be a dissipative particle dynamics, and the links among different links between different particles will be the bonds, which is invisible between the vertices. And to construct a red blood cell with the correct mechanical property, we impose this potential among the vertices. For example, the elastic property will be preserved by this in-plane potential, and then the bending rigidity of the red blood cells will be preserved by this potential. So next, as I mentioned, there are interesting chemical and physical process of thrombus formation. I'm interested in showing you my contribution in terms of mechanics, where the excessive aggregation of red blood cells are stopping the cells from passing this microvascular freely. And another will be the multi-process process scenario where I use a dissipative particle dynamics model and extend it to incorporate both chemical process called coagulation cascade and the physical process of cell adhesion. So let's start with the first one. So many of you may be familiar, the capillary network are shown like this, and it's pretty small compared to the diameter of red blood cells. Therefore, it will have a risk of blockage if the cell cannot freely flowing in the capillary network. And from our clinical observation, we found that the fibrinogen, which is an adhesive molecule, the concentration of which is actually proportional to the red blood cell aggregation, meaning that the higher the fibrinogen is, the red blood cells tend to aggregate together in different patients. So we developed this hypothesis that we can construct these fibrinogen concentration-dependent adhesion models shown by the figure here. So for different patients with different fibrinogen concentration, we constructed different red blood cell models. And we use the fibrinogen concentration to inform the proportion of the blue particles on top of all the red blood cell particles, because the blue particles we here are denoting the adhesive particles. So if you see the graph, you can see that the more blue particle it is, the more fibrinogen, the higher fibrinogen concentration we are trying to represent. And to impose adhesion potential, we use a combination of Morse and Alger potential as shown by these formulas. And to validate our in silico model, we try to compare our result of this in silico model simulation with the in vitro experiments done by our collaborator at MIT. We can find that qualitatively at the doublet level, we can see a good pattern of cell passing through the slits of microvascular represented by the microfluid devices. And then we compare quantitatively for each patient. We can see the disaggregation rate denoted by the dark bars are also matches well between the in silico model represented by the dash bar and the experimental model represented by the solid bar for each patient. So this is a very exciting benchmark for our red blood cell aggregation model, and then further employment of this model has been shown to be valid to predict a blood viscosity in hyperviscosity syndrome, collaborating with our collaborator at Northeastern University, and then by collaborating with our collaborator in Joslin Diabetic Center, we also employ it to simulate the cell behavior in the very irregular geometry represented by the microaneurysm in patients with diabetic retinopathy. And using this model, combining with the red cell aggregation model, we can actually also help calculate the forces due to this aggregation on top of the microaneurysms. And it can help us further predict the rupture risk of the microaneurysm. So second, I want to talk about this multi-process where I simulated both coagulation cascade, the chemical process, and including my previous attempts in cell adhesions from other projects to representing the physical constraints. So as we remind that the important component of the thrombus formations are the blood cell mechanics and also cell dynamics, as well as cell interaction denoted by the adhesion we are focusing on in this talk. And then the second component is the chemical transport of these coagulation factors mediated by this mechanism called coagulation cascade, which can be activated by either intrinsic pathway or extrinsic pathway. And in this modeling, we are primarily interested in the intrinsic pathway activated by this TF7 factor 7A factor 7A complex, meaning that once this concentration of this complex reaches threshold, the extrinsic pathway will be activated. And to simulate this complex intertwine between physical and chemical components, we employed a model called CDPD, meaning by simulating a 21 coupled ordinary differential equation representing the dynamics of coagulation factor, 23 coagulation factors. So different from our original setup of dissipative particle dynamics, now the fluid particle to represent the transport of different chemical chemical species will be extended from only the original 3D vector to a longer vector with almost 23 dimensions. And this is our first in silico model to holistically investigate the microthrombus formation in COVID-19 using the parameters provided by our collaborator from MGH, addressing both cell dynamics of the physical components and also the transport of chemical species representing the chemical component of thrombus formations. We have observed very interesting patterns in terms of the growth rate of the thrombotic at the thrombotic site. So the growth rate we are interested in looking at is the number of platelets adhere in a fixed time period. For example, from this graph here, you can see the x-axis denoted blood velocity, and the y-axis denoted the growth rate of the platelets at the thrombotic site. So the first conclusion we obtain is the flow velocity we found the flow velocity dependent promotive than prohibitive platelet aggregation pattern, which correspond very well to the mouse study done in 1970 for different thrombus for different complex concentration levels. And also the critical flow velocity for the ankle point, for the elbow point or knee point of the phenomenon to happen, which denotes by the circles for different curves in different colors. The critical velocity of this phenomenon also happen to increase, meaning that with respect to the higher complex concentration. Meaning that if you look at the elbow point, the y-axis, the projection of the elbow point for the blood velocity will increase when we have a higher the complex threshold in the initial concentration. So those are my work done in multiscale modeling. And why am I interested in machine learning though? Is that from my previous attempts, I found that if you want to simulate the same phenomenon but at different time level or scale level, you always suffer from the lack of computation and resources. So for example, traditionally, the multi-scale model at different levels are represented by these have some representative method like this 4 method. So machine learning will be help to mitigate these challenges by interfacing between different time scales with some noticeable attempts in the past. And our focus in my future lab, in my lab right now, is the physics-informed neural network as well as the neural operators to help us understand the pattern among different scales better. So I want to talk about my previous attempts to develop machine learning models, specifically focusing on glucose predictions with deep learning. So I guess I don't need to mention the motivation of patient-specific glucose prediction in diabetes is that it's important because when in low glucose, patients will suffer from coma or hospitalization, whereas in high glucose, it will develop further risk of cardiovascular diseases. Therefore, accurate glucose prediction is a critical process for decision making. And on the left is a more apparent motivation of this is that real-world diabetic glucose for 40 patients over time look similar, yet also different in terms of their own patterns. So we can observe that glucose trajectory for diabetic patients are highly variable, and also there are inter-patient heterogeneity, meaning that the curves are not following exactly the same pattern due to different lifestyle or genetic reasons. Therefore, to develop these glucose prediction algorithms, we require a timely and accurate models, specifically taking into account of patient-specific predictions. However, given this very desperate real-life observation, we are faced with nonlinearity of the model, and also because of timely prediction of the fatal, say, low glucose level, it gives us really a small data challenge, meaning that if we want to develop patient-specific model, we are suffering from small training data, and also the timely also impose the same problem of small data for our model training. So to solve these challenges, we follow the traditional way of defining a data-driven glucose prediction model, is that we frame this problem as using the input to be the past 30 minutes of glucose trajectory for at different time at every five minutes. So basically 7-dimensional data input, and then our output will be the future glucose at 5 minutes or 30 minutes or etc. Okay. So recall from the challenge of nonlinearity and small data, we want to tackle the first challenge by testing, developing a plurality of neural networks from recurrent neural networks to convolutional neural networks and to transformer. Meaning that we are using specialized neural networks which capitalize the time series patterns in our data. And then for the small data, we employ a technique called transfer learning. And specifically, because we have a bunch of patients' data, we use this strategy called leave one out transfer learning. And it's being and the following approaches are being used is that the following steps are in this transfer learning is that first we train a very pure model on all but one patient's glucose trajectory data. And then in the second training step, we'll fine-tune using 4 different methods on fine-tune the model on a targeted patients using just a little bit of their training data. And this idea actually also helps develop the recent very popular foundation models in different areas. So it seems that we can tackle the second challenge of small data using transfer learning. So how about the results? So our approach has been shown by a lower error in terms of both mean absolute error and the root mean square error, and in different prediction horizon, meaning like how further away in time we want to predict, and comparing on our result, our model performance on public data sets, and comparing with those models which have the similar setup yet use a different models for predictions. And if we further zoom into a classification problem, specifically detecting the hypoglycemia, which is very critical, and comparing our results to the like second best or similarly best model, we can find our model actually have a much higher sensitivity in 30 minutes prediction, and actually shows a better results. So in summary, I've presented to you glucose prediction using the continuous glucose monitoring combining with neural network and transfer learning. So what's next steps in my lab is that I want to address 2 factors I haven't considered about. First is the uncertainty quantification. For example, how confident I am my model is in terms of the glucose prediction. And second is the multimodality nature of diabetic management. For example, nowadays with the prevalence of Fitbit or Apple Watch, you can have a lot of input like meal intake, exercise intensity, and which can be further incorporated into the model to improve the accuracy. Okay, Second, I'm very interested to show you my effort in developing patient-specific control algorithms for specifically type 1 diabetic. And just a brief review of type 1 diabetics is prevalent. It affects mostly kids. And then it's usually considered as genetic or infection. And patients suffer from few or no insulin due to the misfunctioning of the pancreas. And since the patients are young, they have to take food to increase their glucose for energy consumption, yet they are active because they are young, and their activity will actually decrease the glucose. And this adversarial relationship is what we are interested in investigating. And current technologies help type 1 diabetic patients is artificial pancreas model, which consists of the following 3 steps. One is automatically monitor glucose levels. Second is calculate appropriate amount of insulin. And third is to deliver the insulin into patient's body. And the wearable technologies for type 1 diabetics patients are shown by this sketch. So for accomplishing the first task will be the continuous glucose monitoring that's being used in the previous study. And then the insulin pump model will be developed to incorporate to deliver the insulins. Therefore, the remaining challenging task is how can we optimize, for the second one, optimize glucose dosage, considering the adversarial relationship between meal, which increase our glucose, and exercise, decreasing our glucose. And specifically, we want to also follow in the same trajectory of considering patient specificity as we seen from the last slide, last project. And to design these algorithms, for me, being a purely computational scientist, I need to separate this goal into 2 steps. One step is to construct an environment to evaluate the algorithm, because I don't interact with patients, or it's not actually safe to interact with patients for their safety consideration. And the second step is to develop algorithms to prescribe insulin given past glucose in a period with the consideration of the limitation of our current technologies, wearable technologies. And specifically, the following work has been done on top using the data collection called Ohio T1DM dataset. So let's first focus on the first step of constructing the environment, specifically glucose insulin dynamics, using only the CGM data. So currently, popular method of reconstructing these dynamics can be divided into, for example, data-driven models using autoregressive models or recurrent neural networks. And then some others fall into the category of knowledge-driven models, which abstract our body into compartments. And these compartment models actually represented by some ordinary differential equations. However, none of it, not every model is the best. For example, data-driven models have suffered from lack of interpretability, and also data-driven model have to be updated regularly to addressing the progression of disease or improvement of therapeutics. Whereas the knowledge-driven models assume a smooth function for our patients, and then it's really hard to incorporate those wearable data, which is actually very noisy due to instrumental errors. So my future outlook for this model is to combining both data-driven and the knowledge-driven model and fall into the category of hybrid model, which represented by neural networks plus the compartment model, as well as data like the data from our wearable data. So this is a brief diagram for the compartment model that I'm interested in because incorporate those factors like exercise and food intake, as well as the insulin pumps that I'm trying to prioritize in the model development. And then the observable state of interest will be glucose as well as unobservable circulating insulin or functioning insulin that I termed. So this is the ordinary differential equation, abbreviated ODE, for the compartment model that from the last slide. And our goal is to take the glucose over time to get the states that cannot be measured states, meaning the value of those variables, which cannot be measured by current wearable technologies. Also, what's more interesting is that we want to also obtain the parameters that's governing the ordinary differential equations. And the second goal is to have our patient-specific model, meaning that each patient should have their own parameters as shown by this lump of parameters. And specifically, I am interested in using inverse neural networks to solve this, to address our goals by solving using a mathematical technique called solving the inverse problem. And I want to briefly introduce the solve inverse problem is a way to infer the hidden parameters and hidden states governing ordinary differential equations with partial observation. And in our case is that we are using the partial observation of glucose to infer the circulating insulin and functioning insulin cannot be measured by current wearable technologies. And the way I'm interested in using neural networks is that, first of all, it's good to approximate complex patterns as we observe from the CGM data. And second, because it has auto differentiation with back propagations, which are very useful for solving differential equations. And on the right, you can see the architecture of our systems biology informed neural networks, which takes into the time and then scale it into a non-dimensionalize the time, and then we build a different feature layer to incorporate the models at for different features, and then output the model into different states. So basically, this only boils down to train a feed-forward neural network, which gives out, given a time, will predict the different states in our ordinary differential equations. And by minimizing the loss function, which is a combination of the data loss denoted by the wearable CGM data and residual of ordinary differential equations, which meaning the physics informations, and third is initial value where we want to incorporate to make the system is more stable. Oh, sorry. And the key novelty in my work is that I use adaptive weights for each loss. So here you can see there is no coefficient for different loss term, which is like a coefficient is 1. And we found that by having adaptive weights for each loss, it allows for faster convergence, and then also the time-dependent hidden parameters. We modify the model such that the parameters will be changing among time to account for a long-term glucose variations, which cannot be characterized. Okay, so now I'm showing you how good the result is. Like taking one patient, we can see a good collapse between observed state with other inferred forward and predicted models by our neural network as well as the parameters. And then we assume the insulin functioning are also inferred very well. The shaded area are representing the average of 5 different rounds. Okay, now that we have a model that can be used to test the algorithm, how can we develop the algorithm to control our glucose? I'm interested in automating this insulin infusion dosaging using offline reinforcement learning. And before I go to offline reinforcement learning, I want to introduce the reinforcement learning, which is a technique to train an agent through trial and error, and then to perform optimal decision making. So when, for example, taking this toy problem of training our dogs to perform sitting, right? So maybe at one attempt, the dog will be wandering around, and when you say the command of sitting, the dog will be wandering around, and it accidentally perform a sitting, and we will reward it with a meat. And then the second, and this is a discounted reward in this training attempt. And in second attempt, our dog maybe the other day find out that when it performs sitting, it will quickly get meat, so it actually reduce the corresponding return. And then in the third attempt, the time elapsed between our commanding and its sitting will be smaller and smaller due to a reduction of the return. I mean, with a reduction of the return. Sorry, increase of the return actually. Yeah. Okay, so going back to our problem, when we want to automate the insulin infusion in real time, we're actually taking the past glucose because that's mostly the wearable data we can acquire, and then we train these robots algorithm to optimize glucose insulin dosage and to stabilize our glucose. The challenge of this study, or actually all biomedical data study, is that we need to avoid this the consequence of errors due to the in the training step, and specifically for me, I want to avoid the low glucose, which may lead to coma during this training process. And one way of tackling this challenge is using a technique called offline reinforcement learning, where the agents for insulin dosage no longer interact with the patient, but actually interacting with a buffer containing the past glucose and insulin usage of that patient. So we systematically tested two different offline reinforcement learning, and we evaluate the performance of trained agents using patient-specific glucose insulin models that we developed in the first attempt. And we found very interesting observation is that first, offline reinforcement learning, the insulin dosage performed by offline reinforcement learning could outperform the data it's being trained from. However, we also find there is no free lunch because the quality of the offline data is very important in deciding whether the training will be successful. And third is we found that domain knowledges could help improve agent model performance. Specifically, I found that expanding the insulin action search space for patients who have higher glucose from the offline data collected will help the offline training of the reinforcement learning. So in summary, I've shown you how I can construct digital models using physics-informed neural networks, or specifically systems biology-informed neural networks, using all the differential equations and variables and neural networks. And then also show you attempt to automate insulin delivery with a better glucose stability. So what's next step or outlook of our model is to maybe taking into account more realistic, like the reaction rate of insulin, because now in our prototype, like a primitive models, we didn't even consider like there is a lag between insulin delivery and insulin action. And more specifically, if we are interested in understanding how the spatial variation, which can be represented by the partial differential equations shown here, and then using that, we can maybe understand how the spatial temporal dynamics and even different drug routes will affect for patients with diabetes or any other disease. Okay, so here is the last project on analyzing high throughput biology data. So I don't need to mention like how devastating to see the coronavirus has affected so many patients and also leading to so much death. And then we also know the molecule protecting us from COVID or any other infectious diseases is this molecule called antibody. And typically, when we measure the antibody potency or functionality, we focus on an assay called neutralization, which only analyze the antibody binding fragment of the FAB region denoted here. However, some recent studies have shown that the tip part of the antibody, which is called non-neutralizing Fc functioning antibody, can also be protective in some HIV clinical trials. Therefore, it remains to be understand how to evaluate these Fc functional antibodies. And we focused on the recent techniques developed by my postdoc lab called systems serology. It specifically take all the serum of different patients and then I mean vaccinees to analyze the diversity of antibody using antibody biophysical profiling and understand the function of antibodies using this antibody function profiling. And for me, this is just a lump of data which has a higher dimension yet a lower sample. This imposed to me the challenge because this high dimension of measurement and low dimension in terms of patient or subjects are very detrimental for a machine learning task because it may lead to overfitting. And then also when we are faced with new viruses, it actually gives us a limited immunology understanding of the disease. Therefore, we design a linear model, which is a low-order model, which decreases the overfitting, and those coefficients that we shown here by this linear model are actually meaningful and also interpretable. So typically, I developed these machine learning pipelines to help distill the novel immunology. And the goal will be which system serology features will help distinguish, say, a group P1 and a group P2. So first thing I do is taking this fat data, I do dimension reduction to decrease overfitting. And second, using the reduced dimensions, I project them to a 2D latent space to visualize the separation. And third step is interpretation. I take the coefficients of the features and the corresponding projection in the first latent variable or further if necessary. And these are some examples of the specific algorithm to be used in each steps. So I'm showing you how we can use the system computational the machine learning approach to understand the system serology data obtained from this hybrid immunity in SARS-CoV-2. Specifically, our cohort are separated into two. One is called hybrid immunity, meaning the patients has been infected by COVID-19 before there's 2-dose vaccinations. And then there are a bunch of patients categorized as vaccination only, meaning that they have not seen any viruses before they take the vaccines. And then here is a brief structure of SARS-CoV-2. We want to analyze the antibody responses with respect to different components of the SARS-CoV-2. And specifically, we also examine two different vaccine platform, which is mRNA vaccine and viral vector vaccine. So what we found that machine learning is very helpful in distinguishing antibody signatures in different groups. For example, you can see in the both platform, our separation of vaccination group denoted by the blue colors and hybrid group denoted by the yellow colors are pretty good. And more importantly, the projection of the coefficients for the machine learning model shows us the hybrid immunity actually triggered more S2 specific immunity in both platforms. And if we go back to the structure of SARS-CoV-2 viruses, we can see that S2 is a conservative part of the virus. So the same systems biology platform, systems serology platform, and the corresponding machine learning pipeline has shown to be very useful in both understanding how the vaccination route in influenza affect immunology, and also how protein vaccine responses against SARS-CoV-2 works in non-human primates, as well as understanding the immunology changes in mRNA vaccination against SARS-CoV-2 in kids. So in summary, I've shown you how a linear model can be used to analyze this high-dimensional omics data for antibody profiling. And what's the next step is that a graph model, which embedding a more high-order interaction among different entity of our immunology data, will be considered very interesting to me. And then also because due to my previous understanding of the ordinary differential equations, I want to also incorporate my previous expertise in ordinary differential equations to see if we can actually add more causality of the interactions in either both high order and the linear order model. So, I mean, I want to also briefly talk about my future plan, also my current plan of my current lab is that we're interested in integrating machine learning and multi-scale modeling in both medicine and engineering. Specifically, we had the following active projects where we develop efficient deep learning models for biomaterial discovery and scientific machine learning for glucose managing in diabetes, and physics-informed machine learning for engineering designs like batteries, plastic, or biomolecular designs. With that, I want to conclude my talk, and thank you so much for your attention.
Thank you so much, Yixiang. This was absolutely fascinating and to the future.
Thank you.
Any questions for Dr. Deng? Well, while we are waiting for questions, I have a couple. I guess when you mentioned the solve inverse problems, I was not quite sure I understood there because I mean with the model, which is absolutely amazing to be able to predict to have an artificial pancreas.
Yeah.
Including exercise, including all this. I mean, it's totally amazing. What you want to do, if I understand well, is to provide insulin before the glucose raises, or stop insulin going before the glucose goes down because of exercise, for example. And so, but by using glucose as as a way of, I'm not sure how I can say that. It seems that here in your model, the solve inverse problem, right? Glucose there is the marker for the production of insulin, right? But wouldn't you have insulin production before even the glucose, goes down or up? I mean, I guess physiologically the insulin reacts to the glucose level. That's true.
Yeah. So let me understand your question. So you are not sure about what's the solving inverse problem is here is working.
Right. How does it work?
Yeah. So solving inverse problem generically for like ordinary differential equation, like differential equations are given, say, the observation of states. For example, the states are the insulin over time. We have a comprehensive observation of all these states, assuming that. But we also know how our system looks like, but we don't know the parameters that's governing the observation. Then we solve the inverse problem because we want to preserve the dynamics. I mean, the solution of our inverse problem should preserve the dynamics that we obtain from the sensors, right? And then you also should preserve the structure, like the dynamics between IXG looks like this, but we just don't know how much this value is, this value is, this value is. And the inverse problem is trying to help you achieve this, like to determine the value of these parameters.
Wow, that's really, yeah, that's fascinating.
Thank you.
So where do you stand in your glucose pancreas, the artificial glucose? What do you need and where do you stand now?
Yeah, I mean, like the progress of this
Right.
research.
Yeah.
Yeah. So we actually receive a lot of feedback in terms of medical in the medical side, actually, because one thing is there are established even like FDA approved on the differential equations, which is not the differential equations that we use here. So some people will ask like, why don't you use that? And then that's one, I mean, that also drastically changes. We need to redo everything using a different set of ordinary differential equations. So we are still figuring things out. And then also because we are capturing a really long time trajectory, if you can see here from December 1st to January, it's like 8 weeks. I mean, the data we had for that the public data said Ohio T1DM follow up with 2 months, like 2 months, like 8 weeks of this data. So we also receive feedback saying that the time is too long for the given all the differential equations that we focus on. So we might also need to do some that kind of changes to adapt to the reviewers' comments.
I see. So will you still use this database, the Ohio database, to do that?
Yeah. I think so. We probably will focus on that because, or yeah.
I see.
But we welcome more data because that data was acquired in 2018, so it's almost 7 years ago. And if there is new technologies with more comprehensive data, we are looking for those too.
I see. And I mean, you have a lot of data in this database, but it's a small number of patients, right? How many patients
Yes,
do you have? It's
6 patients.
six
Yes, you're
patients,
right.
right?
Yeah.
Who are followed up for 2 months.
Exactly. Actually, that's one of the rebuttal comment we got from it, but we argue because it's patient specific, right? So sometimes you can get rid of, and also we even did a sensitivity analysis. 6 is roughly the limit of a sensitivity analysis.
Right.
But yeah, we still encountered a lot of challenges from the reviewers.
I see.
Yeah.
All right. Any other questions? Oh, it seems everybody is must be fascinated. Well, thank you so much. It's the top of the
Yeah,
hour. Thank you so
of
much
course.
for this really